Hello, my name is Alexa Knight. I am a junior in the biomedical sciences major at the University of Washington, Tacoma, and with the support of Dr. Alaei, we have been identifying and characterizing arsenic-resistant bacterial species from periphyton communities. The source of the bacteria in our study came from periphyton samples. Periphyton is a community of different organisms including blue-green algae, bacteria, fungi, plant detritus, and animals. In addition to providing dissolved oxygen and soil to its environment, periphyton has been shown to accumulate arsenic at a far higher concentration than the water around it. The periphyton samples were provided by Dr. Jim Gowell, whose team is studying arsenic uptake by periphyton, and had ruled out algae as being responsible for this uptake. Our lab aimed to discover if bacteria were in fact responsible for the uptake of arsenic observed. We isolated bacterial colonies from the periphyton and ran 16S rRNA sequencing to define what species of bacteria were present. Much to our surprise, we also found that our bacteria grew readily in conditions where the arsenic concentration was 1,000 times that of their natural environment, suggesting that the bacteria within periphyton might be capable of metabolizing arsenic to resist its toxicity. Arsenic contamination is a global and local issue. The Natural Resources Defense Council reports that in 25 reporting states, they estimate over 56 million Americans are consuming water containing arsenic at levels that present a potentially fatal cancer risk. Here in Washington, many areas exceed the 10 parts per billion level safe arsenic consumption, and some even exceed over 50. Everywhere on this map that contains a black spot is an area over 10, and the red dots represent areas over 50. Consuming arsenic at these concentrations has been known to lead to a wide range of adverse health outcomes, including neurological effects, obstetric problems, high blood pressure, and the development of fatal cancers. A novel approach to arsenic bioremediation is needed as current technologies are expensive and inefficient. This is what we kept in mind as we explored the potential for our arsenic-tolerant bacteria. There was much we needed to discover first before we could think about bioremediation applications. First, we wanted to figure out how these bacterial species within periphyton were impacting bioavailability of arsenic. We knew they were extracting it from the water, but after that we weren't so sure. Are they transforming it, pumping it out elsewhere, or just storing it? We also needed to characterize and identify the species that we had. We've established stocks of 21 isolates so far, and we are currently sequencing 16S RNA genes to identify them, but preliminarily we believe that we have species of Pseudomonas, Genethobacterium, and Rhodococcus. We aim to establish a mutant library to pinpoint the genetic source of our arsenic tolerance. Additionally, we wondered if and how arsenic exposure was affecting biofilm formation by our isolates. Currently in our work, we have successfully introduced a transposable element into one of our isolates. Next steps now include generating a collection of insertional mutants that we can screen for the loss of arsenic tolerance, in hopes that we can narrow down the genetic source. Identifying this source has future bioengineering applications, as it is possible a novel approach for bioremediation of arsenic contamination could be developed from this source. Initial findings in our biofilm assay have qualitatively shown little difference in growth from samples exposed to no arsenic as compared to those exposed to 1,000 times the amount of arsenic present in the lake they were sampled from. It is surprising the sheer amount of arsenic these isolates are able to grow in, and further analysis will hopefully show more clearly as to how the arsenic is impacting adhesion and biofilm formation. There are still many research avenues left to explore with these isolates. We will continue to build our mutant library to isolate the genetic source of arsenic tolerance, and further work will be done in clearly defining arsenic versus non-arsenic growth curves for these isolates. We will also continue to work on identifying species, which will allow us to explore what is already known about their metabolic activities. Finally, we will start silver nitrate assays, a method that can be used for screening collections of mutants for the loss of arsenic oxidation. It can also be useful for the screening of libraries to investigate genes involved in oxidation or reduction of arsenic. I would like to thank Dr. Alaei for her continued support and guidance on this research, Dr. Jim Gowell and his team for providing us with our preliminary periphyton samples, Alexis Heath for her work, the Sciences and Math Division at the University of Washington, Tacoma, Dr. Alaei's microbial physiology class, and the Mary Gates Research Endowment for their financial support of me and this project. Thank you.